Hello, this is Alicia Callow. I work for the University of Illinois Extension as a horticulture educator in Southern Illinois. I'd like to welcome you to our second part of our seed starting module within the Community Gardening webinar series. In this section, I will be talking about germination, the process of germination and the conditions required, the supplies you will need for seed starting, and several methods of seed starting. To get us started, I'll spend a little bit of time talking about the process of germination. Germination is the first of the 3 stages of starting plants from seed to grow in the garden. Germination is the process by which the quiescent, dry seed imbibes water and begins to grow. In the first part of germination, the seed imbibes or absorbs water from the soil. And then that water in turn activates enzymes that are able to turn the food stores or the starches and oils that are stored in the seed into energy. That energy is used for cell elongation and growth. Because of this energy, growth begins, and the first thing to grow and emerge is the radicle or the primary root. After the radicle emerges, the second portion to begin to grow and expand is the hypocotyl, which is the stem between the leaves and the root. The hypocotyl emerges and then it pulls the cotyledons or the seed leaves up out of the soil. After this stage, the plant begins to grow true leaves, which are the adult leaves. The cotyledons are where most of the food is stored in the seed, and the cotyledons will feed the seedling growth until the true leaves begin doing photosynthesis to produce their own food. At this point, germination is over. In order for seeds to germinate, there are several environmental conditions that must be met. The first of these conditions is water. As I mentioned before, the first step of germination is for the seed to imbibe water. This means that we must provide an even moisture in the growing media in order for this to occur. We do not want to allow our growing media to dry out at any point during germination. The next condition is oxygen. Seeds do require oxygen in order to germinate. The main concern with regard to oxygen is not having waterlogged growing medium, because a waterlogged soil has no oxygen. The next condition is heat. Depending on the plant species, seeds will germinate at different temperatures, but most species will grow between the range of 65 to 85 degrees Fahrenheit. You'll have to check out information about the specific species that you're growing to know what soil temperature you should be targeting. The next factor is light. There are some plant species whose seeds require light in order to germinate. A few examples are lettuce, eucalyptus, and coleus. When we're planting these species that require light, typically their seeds are sowed directly on the top of the soil media and are not covered. Other species require darkness in order to germinate. Some examples are sweet pea, phlox, and vinca. These seeds will typically be buried below the media surface in order to be in darkness to germinate. Now that we know how to create a favorable environment for germination, I do want to mention seed dormancy. Some seeds have a condition called dormancy that prevents them from germinating even when they're in a favorable environment. The 2 most common methods for breaking dormancy are scarification and stratification. Scarification essentially uses a mechanical method to break down a hard seed coat. Some of these methods include abrasion, an acid soak, or a hot water soak. Stratification provides a cool treatment which will typically change some of the physiological characteristics of the seed. This can be in either a moist or a dry condition. Now, if a species exhibits dormancy, typically the seed packet will provide instructions on how to break dormancy or how to promote germination. If there aren't specific instructions on your seed packet, you may have to do some research. But for most garden vegetables, this will be taken care of already. Now that we have established the conditions needed for germination, I will go over the supplies that you'll need for seed starting. First thing you'll need is a germination or seed starting media. You'll also need containers for your media, such as seed trays, flats, or plug trays. You'll also need to consider having a water supply, such as a fine bulb sprayer or a bottom tray to water in. You'll also need a heat source that will be able to provide heat to the bottom of your container. In order to have good success at seed starting, you'll want to select a high-quality germination media. You'll want to select something that's a soilless media mixture with a light, fine texture. Typically, the ingredients in a seed starting mixture will include sphagnum peat moss and vermiculite. Your mixture should be low in soluble salts or fertilizer, so look for something that does not have an added fertilizer already in it. You'll also want to find a sterile product. The goal of a seed starting mix is to have something that will hold water, but not too much, so it will still have good drainage. This is to prevent waterlogging. An alternative to using a loose mixture is to use peat pellets. Peat pellets come compressed and dried, and once they're moistened, they expand. Peat pellets are still typically placed in a secondary container, such as a flat or plug tray. They can also be just placed in a tray without any divisions. Now I will discuss 3 methods of germination based on the different types of containers that can be used. The first method is using a seed tray. When using a seed tray, a fairly small container is filled with germination media, and the seeds of all one type are planted together in this small tray. Then this is placed on our heating element and allowed to germinate. Once our seedlings have germinated, the strongest and healthiest are transplanted into a larger container, such as a flat or an individual pot. The advantage of using a seed tray is that the seed tray takes up less space than a flat on our heating element. It also allows us to pick the seedlings that we want, the ones that are healthiest and largest, and it saves space in our final flats from having little weak seedlings in some of those individual cells. The disadvantage to using a seed tray is that some seedlings do not tolerate having disturbance early on in their life, or in other words, they'll die due to transplant shock. It can be difficult to disentangle the very tender roots and stems of young seedlings at this stage, and so transplanting can be a difficulty. The next type of container that can be used for germination is a plug tray. A plug tray is somewhere in between our final size container and a seed tray. The advantages of using plug trays is that it allows us to directly seed into individualized cells as opposed to having all of our seeds together in a container such as in our seed tray. This allows us to minimize our early root disturbance because we don't have to separate those seedlings as in the seed tray. The disadvantage to using a plug tray is that they can be more expensive to purchase and will require more space on our germination heat mat. And so this will limit the number of plants that we're able to germinate. The nice thing about using a plug tray is that it's easier to separate out those plants when we're ready to pot them up into the next larger size because those roots are contained in an individual cell. We may still have to pot up one more time before our final container and transplanting into the garden, but depending on the size of our plug tray, we may not have to and may be able to move directly from the plug tray to the out of doors. The final type of germination that we can use is directly seeding into our final container. And this could be our flat of a large enough size that our plants will get big enough before we have to transplant them to the garden, or an individual pot. The advantage of using this method is that we won't have any root disturbance until it's time to transplant into the garden. It's also nice because we don't have to do any transplanting other than directly from the container we put the seeds in into the garden, so we only have to use one container. The disadvantage to doing this is that we might not get uniform germination in all of the cells because seeds do vary in their ability or viability to germinate. This also causes us to waste space in our germination area because we'll have our full-size flats and pots on our heating source. It also requires us to plan ahead for the potential for not all seeds to germinate. This means that we'll have to use more seed than we would have if we were using a seed tray. So we'll have to plant 2 or 3 seeds in each cell and then come back and thin if multiple seeds come up in an individual cell. Once you have decided which type of germination containers you're going to use, you're ready to begin sowing seeds. I'll go through the steps for sowing your seeds. The first step is to pre-moisten your germination media. The reason that it's important to pre-moisten is because sometimes peat moss can actually be resistant to taking up moisture when it's completely dry. So pre-moistening will avoid the unhappy condition of having your pots or flats full of media and then watering them and having it all float away and overflow into a big mess. Once you've pre-moistened your germination media, your goal is to have a mixture that is lightly damp, not soggy. You want to think of a sponge once it's wrung out, not when it's still dripping. When you have your media at the correct moisture, fill your containers and tamp it down lightly. You don't want to tamp not at all, because once you water again, that media will settle and you'll end up with containers that are only half full. However, if you overfill your containers and squish them in too tightly, you'll have removed all of the pore space, which is where the air or the oxygen is that's important for germination, and you'll also decrease the ability of your media and your pots to drain. Once you've filled your containers with media, it's time to sow your seeds. The seed packet should tell you the depth at which to sow your seeds, but if it doesn't give you any instructions, a general rule of thumb is to place the seeds at a depth equal to 4 times their diameter. For small seeds, it might actually be more practical simply to sow them directly on the top. You'll want to pay attention to whether or not it says to cover your seeds over or whether to leave them directly on top because of light requirements of some seeds. If the seeds are to be covered, gently firm the media over the top of the seeds. If the seeds are not to be covered, go ahead and lightly press them into the soil surface so that they don't wash off when you water. Once you have your seeds planted, you want to make sure that you label the container and include the date. This will allow you to keep track of when you should expect to see seeds germinating. And it will also help prevent you from forgetting which plants you have where. Depending upon the size of your seeds, there are a couple of different approaches that you can use to sow your seeds. Large seeds are fairly easy to be planted by hand because they're large enough to see and pick up with your fingers fairly easily. You can simply pick them up and place them in the container at the appropriate depth. You can also tap large seeds out onto the soil surface and then poke them with your finger down to the appropriate depth. For smaller seeds, they're a little bit more difficult to see and pick up with your fingers, so there are a couple of different methods that you can use. First is to broadcast small seeds with a carrier. To do so, mix a clean teaspoon of sand into the seed packet and mix thoroughly. Then you can broadcast that seed over the container in a crisscross pattern. This means that you'll put half of the seed over the container going in one direction, and then the other half you'll put over the container in the perpendicular direction. There are also seeders that you can use with small seeds. There are a couple of different types of seeders for use with small seeds. On the left, you can see a wand-type seeder, which is essentially a bulb that you can press, which will remove the air from inside that bulb, and then you'll place it over the seed you want to pick up and release the bulb, which will create a vacuum that will pick up that seed to the tip of that wand. Then, once you have placed the tip of the wand in the location you want the seed, you squeeze the bulb again and it releases the vacuum. The other type of seeder is essentially like a vibrating trowel. And so the seeds are placed in a reservoir toward the top, and then there is a baffle which has a little hole or opening, and there's various sizes for different sizes of seeds. The vibration causes the seeds to pass through that hole one at a time, allowing you to space them out. After your seeds are sown, place the container underneath of a clear dome or plastic. If you're using plastic, make sure you use some sort of support to keep the plastic off of the top of the germination media by a couple of inches. You don't want it to lay directly over the top of your container. The purpose of using this covering is to maintain the moisture in the media. You don't want it to dry out too quickly. During your period of germination, you will have to maintain the moisture in the germination media by watering. This can either be done by watering over the top or by watering from the bottom. If you're going to water from the top, you will need to have a fine mist nozzle. This is because most regular sprayer nozzles for a hose or from a watering can will be too strong of a water stream and will move the germination media and either change the depth of your seeds or wash them completely out of the container. You need to be able to use a fine mist that will gently water over the germination media without moving it from its place. In order to avoid having to purchase a fine mist nozzle, you can also just water from the bottom. You can take your container and place it in a tray of water and allow it to soak up some water for a few minutes. Don't leave it standing in water for a very long time, especially not all the time. This will cause your soil to become too saturated or waterlogged. And this will mean that there won't be enough oxygen for your seeds to germinate. There are also some other problems that come along with having your germination media too wet, such as promoting algal or fungal growth and by attracting spore flies or fungus gnats, which are both insect pests common to germination. One of the most critical environmental conditions that you can control to create a favorable germination environment is temperature. Note that the critical temperature for germination is not the air temperature, but the temperature of the germination media. You can control the media temperature by placing your containers on a germination heat mat with a thermostat. The thermostat is connected to a probe placed in the media to regulate its temperature. You can see an example of a thermostat probe toward the back of the picture of the purple pepper seedlings. These heat mats are designed to be water resistant so that they're safe for use without the threat of electrical shortage. Do not use a household heating pad as these are not water resistant. The ideal temperature for germination varies among different plant species, but the general range is between 65 and 85 degrees Fahrenheit. Cool season crops such as broccoli, carrot, lettuce, and onion germinate best at cooler temperatures between 40 and 60 degrees Fahrenheit. Depending on your germination space, supplemental heat may not be required for these crops. Warm season crops such as peppers, beans, and melons germinate best at warm temperatures between 70 and 85 degrees Fahrenheit. These will almost certainly require bottom heat in order to germinate well. The final environmental factor that you'll need to consider for germination is light. Most seeds do not require light for germination, and some, in fact, require darkness. Most of these seeds are covered underneath of a layer of germination media or soil, and therefore they are already in darkness. There are, however, some seeds that do require light to germinate. Some seed packets will explicitly say that the seeds require light to germinate, while others will use an essential sort of code saying do not cover the seeds. This essentially is code for these seeds require light to germinate. Some examples of seeds that do require light to germinate are lettuce and eucalyptus. Once you've planted your seeds and placed them in the appropriate environment, they will begin to germinate as long as they are viable and are not dormant. As more and more seeds begin to germinate, at some point you will have to make the decision of when to transition from germination phase to seedling growth phase. It's best, in order to make this decision, to have an idea of how many plants you will want finally in the garden. When you have this target number, wait for 20% more of your seeds to germinate than what you need. This is because not all of your plants will survive through the process of seedling growth and then on to transplanting into the garden. When you're ready to transition to seedling growth, you'll be removing your germination trays from their heat mat, removing the plastic cover, and putting your seedlings under lights. Seedling growth requires different environmental conditions than germination. Some of the factors that are important include light, the growing media, water, fertilizer, and the temperature. These factors, as well as some common problems that can occur during seedling growth, will be discussed in the next section. This concludes part 2 of our seed starting presentation. Up next, part 3 will cover seedling growth. For more information about seed starting and other horticultural topics, visit the State Horticulture page on the University of Illinois Extension website. If you have any questions about this presentation, feel free to send an email to either presenter, myself or Martha, at our email addresses as listed.